Namaskar. Today we'll study the difference between gene, DNA and genome. So we all know DNA, deoxyribonucleic acid. It is a helical structure with two strands, and this is DNA. Why it is there in our body? Because it carries all the hereditary information. And from DNA there will be a process called transcription, by which there will be production of messenger RNA, and from messenger RNA then there will be translation and we will get the proteins. The total DNA in our cell, in each cell, whatever total DNA is present, that is called the genome. And gene is a part of that DNA. So as I said, from DNA we know messenger RNA will be synthesized, then from messenger RNA, in presence of a transfer RNA, we will get these proteins. So a gene codes for a protein. So gene is nothing but a part of DNA which can code for a protein. And if you see the structure of DNA, it is a chain of sugar phosphate, sugar phosphate, sugar phosphate. So here if it is DNA, then it is deoxyribose sugar, and to each sugar there is a base linked, and these base can be G, A, T, C, guanosine, adenine, cytosine and thymine. And there is cross linking of G with C with triple bond and A with T with double bond. So when this cross linking is there, we get this double helical structure, and we can know the length of a DNA in terms of its base pairs. So this is one base pair, this is one base pair, these are the two consecutive base pairs, and if we see the distance between two consecutive base pairs, it is around 0.34 to 10 to the power minus 9 meter. So between two consecutive base pairs, this is the distance. And what is the number of base pair in DNA? If you see this DNA in our cell, it is present in the nucleus. DNA is also present in mitochondria. So in our case, in animals, in human beings, we have DNA, nuclear DNA and mitochondrial DNA. In plants and green algae, there can be further chloroplast DNA also. So when we say genome, that means nuclear DNA, mitochondrial DNA, chloroplast DNA. In our case, we will say genome, nuclear DNA and mitochondrial DNA, because we don't have this chloroplast DNA. And if we see this mitochondrial DNA is very less quantity, but this nuclear DNA, again, this DNA is present in the form of chromosomes. So chromosomes in human beings, we will say the 23 pairs of chromosomes are there, and 23 chromosomes, haploid gene will have 3.2 to 3.3 billion base pairs, and total it will be 6.6 billion base pairs. So 6.6 billion base pairs into this one, so it will be around 2.2 meter in length. So you can see that what is the length of this one DNA per cell is 2.2 meter, and 2.2 meter length DNA is inserted into the nucleus, and the nucleus size is only 10 to the power minus 6 meter. That means this DNA is condensed in the form of chromosomes, chromatin fiber inside the nucleus. And a part of DNA is our gene, which is having around 100,000 base pairs. A gene is having 1 lakh base pairs, and total nuclear DNA will have 6.6 billion base pairs. And total DNA in our body is our genome, and in human genome we have almost 20,000 to 25,000 genes. To be exact, I think 26,000 genes are there. Number of genes will have 26,564 in human genome, and 26,564 to be exact number of genes in genome. So this is the difference between gene, DNA and genome. So gene, to summarize, gene consists of enough DNA to code for one protein. DNA is the hereditary material in on all living cell, and genome is the sum total of an organism's DNA, which includes your nuclear DNA and mitochondrial DNA. So it is the entire set of genetic instructions found in a cell. So it includes the chromosomes in the nucleus, so 23 pair of chromosomes. So 2 into 3.3 into 10 to the power 9 base pairs. So 3.3 into 10 to the power 9 is your haploid genome, and diploid genome into 2. So it will be around 6.6 into 10 to the power 9 base pairs. And a small chromosome is also found in mitochondria, which is having only 16,500 base pairs, very small, and it contains around 37 genes. So you can say our genome is having almost 6.6 into 10 to the power 9 base pairs, or 6.6 billion base pairs. And all these are inserted in the nucleus, which is having only 10 to the power minus 6 meter size. This nucleus is having only 10 to the power minus 6 meter size. So it is inserted, this genome is inserted because of this condensation, and the condensed form of this DNA is nothing but your chromosome. So this is the difference between gene, DNA and genome. Thank you.